Hello, and a warm, warm welcome on this frosty day. Thank you all for joining us for the third annual Senate Forum. My name is Grit McCreath, Chancellor of USask. Even with this event being held remotely, let us acknowledge that we are on Treaty 6 territory and the homeland of the Métis. We pay our respects to the First Nations and Métis ancestors of this place, and we reaffirm our relationship with one another. The topic of this year's forum is the race against COVID, innovation the world needs. And it couldn't be more fitting for USask experts to speak about this topic. We are global leaders in this area and at the center of this race against COVID, with our own vaccine candidate now in human trials. Not only are we leaders in developing vaccines, but we also have the expertise in community health and how a vaccine can be effectively administered province-wide. Indeed, we are the university the world needs, and you will learn a great deal about these and other topics over the next couple of hours. When I think about how our University of Saskatchewan is solving these fundamental global challenges, I feel a great sense of pride, as I hope all of you do. COVID has changed so much for all of us. For our campus community, the pandemic fundamentally changed the way that we teach, the way we learn, and the way we interact and connect with one another locally and globally. That includes how Senate meets. In time, I know we will be able to hold this forum as well as our regular meetings back on campus and in person. And I truly look forward to that time. As members of University Senate, we each show our dedication to our alma mater. We are ambassadors for USask in the communities we live. And after today's forum, I know we will have so much more to share, remotely of course, about the amazing work happening at USask. So I thank you for your unwavering commitment to Senate and indeed the University of Saskatchewan. Now I would like to introduce Dr. Doug Brothwell, who will be the moderator of today's discussion and introduce our speakers. Many of you will know Doug, who is the Dean of the College of Dentistry, Director of the School of Public Health, and also a member of University Senate. Please join me in welcoming Dr. Doug Brothwell.
Thank you very much, Chancellor McCreath. I greatly appreciate the introduction. I too extend a welcome to all of our attendees of today's third University of Saskatchewan Senate Forum. And without further ado, we will get started. So I thought I would start by trying to set a bit of context about the entire pandemic and what's been going on over the last while. And so what I'm going to do is to start out by just highlighting a few key pandemic issues that have come up since the beginning of the pandemic. And unfortunately, I will use a USA perspective. They have much more interesting news items, headlines, and there's many pictures available. So I'm going to use those three things to highlight a little bit of what's gone on with the pandemic. I'm going to say a few words about the primary role of public health and, strangely enough, about the miscommunication that's been done by the US president. I'll follow that by a little bit of a pandemic toll and then tie that together with how those things relate back to today's forum. So going on, I'll just start with a few news headlines. Here's a clip out of the Seattle Daily Times announcing that churches, schools, and shows are closed and that really there's a ban on all public assemblies. This will not be any news to anyone who's lived in this world. Here's a similar thing from Bemidji, where public places were closed. Here's a headline from Pennsylvania talking about how the pandemic is going through and ravaging things at such a quick rate. And here's a clip from San Francisco where the Red Cross was promoting wearing a mask to save your own life, as it says. And in Portland, obviously an image from early in the pandemic when PPE was in short supply. But here's a clip from an article about wear a mask and stamp out the pandemic. So there's been a lot of headlines, and there's many news clips that you could use to talk about the pandemic. But I'm going to go through now about 6 or 7 pandemic headlines without the newspaper articles, just with the headline itself, and roughly in the order that we might have seen those occur. So the first one is, of course, there is a pandemic that's about to hit the world. It's of unknown origin, but it's certainly putting the world at risk. So we all remember that early in the outbreak. Sort of next after that was some work about, the top scientists are working to determine what the cause of that pandemic is, which virus it is, where did it come from. That, in my opinion, was sort of followed by, what we were able to find out, what our scientists were able to find out, that there was a novel virus that had jumped from animal to human, and that was the basis behind what we were about to face and what we continue to face. Next was some, the attempt to have some good news. So here's the headline where the New York City Health Commissioner is reassuring citizens that there is going to be help on the way and that there are vaccines being developed. To me, the next important one headline was the fact that there are multiple vaccines available, although they are of unknown effectiveness at the time when this headline came out. And that led to certainly what we see a lot in the media now, all of this about the uncertainty about what the vaccine is, how effective it will be, how safe is it, that fueled public doubt. So those headlines are certainly not uncommon. I'll go on a little bit to some pictures. These, I think, tell a bit of a story of the pandemic. So here's an example of how pervasive the changes in society because of the pandemic. So obviously not Canadian, perhaps not US, but a picture of how even the policing that needs to be done has been affected. Public sentiment seems to have been split. We have some people who think that more needs to be done and will wear a sign that says, wear a mask or you ought to go to jail because you're not doing your part. We have other people who are really denying the pandemic and are resisting it. And so here's an example of an individual not wearing a mask and being lectured, obviously, for not having done so. We've all learned new language. One example only is the term flattening the curve. And so this is just an image that shows the efforts to flatten the curve and how effective that was in different areas in the US. Many more, the word variants and are not and a whole bunch of other terms are just new to the public's lexicon now. And of course, all kinds of images of the culprit, the coronavirus, and of course, usually seen in public media, colorized, of course, through computer-based coloration. But certainly not an image that is unusual. And like most generations, every generation needs its heroes. And I will put my opinion forward that our heroes for many, many years are going to be the frontline healthcare workers and other health frontline workers who really have put themselves at the front of the battle against the pandemic. Now just a couple or 3 slides on the role of public health. So I have to do a little shout out to my public health colleagues because we, at the beginning of the epidemic at least, there was no vaccine. And so the control of the damage caused by the pandemic had to be done by sort of more traditional public health measures. That doesn't say that I think they're simple public health measures. They're not. They're just traditional. So certainly the education of the public that is needed to get people to take the right steps to help avoid the spread. There's the surveillance that needs to be done. This needs to be international surveillance, worldwide surveillance, so that public health is able to identify the areas where the pandemic is spreading and the areas that are highly affected, so that the interventions can be tailored to where those are, and that's right down to who within each population is being affected. And as we know, that one of the large target populations is our seniors. One of public health's duties is to predict how soon we're going to surpass the capability of the healthcare system and to put in additional healthcare capacity to take care of those extra people. And of course, I think everyone knows that our very own Merlis Belcher Place has been used as a, sorry, my term, field hospital for the overflow of pandemic cases. And of course, policy. So this is a really bad example of policy. I'll just flip on, but we set up policy that under pandemic conditions often ends up being the law that needs to be followed. Next, I'm going to go on to the US president, and what a strange thing you would think to talk about. But there are two really key headlines here. So the first one being that the US president was responsible for a propaganda machine, that misled citizens and really ended up causing more trouble because of the pandemic than what was necessary. And of course, for those who believe in karma, there is the headline that the US president ended up catching the virus. And of course, here's an image of the US president that I'm speaking of, that led the propaganda machine and who ended up catching the virus. So it was probably obvious before, but it certainly is now that I haven't been talking about the COVID-19 pandemic. I've been talking about the 1918 novel H1N1 influenza flu, which has also been commonly called the Spanish flu. I can't say the word Spanish flu without pointing out that the Spanish flu did not originate in Spain. That is a misnomer of a name for that pandemic, because it is now believed to have originated in the US and specifically in one county in Kansas. But interestingly, the Spanish flu, the 1918 flu, came in 4 waves. About the first half of 1918 was fairly mild. It was not too unlike a normal flu in terms of its mortality and morbidity. But that changed in the second wave. And the second wave was the latter half of 1918, where a new variant came out, and that new variant was much more deadly, had much higher mortality, and uniquely started to affect the healthy in the population, not just the other usual flu targets. Of course, then the third and fourth waves came. They lasted about another 1.5 years after that. And those waves had a slightly lower mortality. Strange thing, I hope one of our virologists can talk about that. It's a process that I understand is called passage, where some viruses will end up being less deadly with time. But that is what happened with the 1918 influenza pandemic. Of course, it's important to put that in context with what was going on in the world at the time. That was during World War I that this pandemic came out. And I think it's noteworthy to mention that the Army surgeon, General Gorgas, made the statement early in the start of the pandemic that this would be the first war where fewer American soldiers died of disease than died of combat. Of course, it would turn out that he was wrong. And that, to me, puts a lot of context in the severity of this 1918 flu pandemic. It was, despite all of the deaths that occurred because of World War I, the flu took more soldiers' deaths than did the war. So the toll of that pandemic, which is, I'm still setting the case for today's presentations. The toll of that pandemic was that it ended up affecting about 500 million people in the world, which would have been approximately a third of the world's population at the time. And it resulted in the death of somewhere between 50 and 100 million people. There isn't complete agreement on this, but it is generally considered to be somewhere in between there. So between 10 and 20% of the people that contracted the flu ended up dying of it. And with almost all flu, the mortality focused itself or was primarily amongst young children and older adults. That is very common, as well as those people with underlying medical conditions who are not able to fight off the disease as readily. But very uniquely, this pandemic affected healthy adults. So it affected soldiers in what would generally be considered the prime of their life. And that did not happen at the beginning of the pandemic. It was an event that came up later and then subsided to some degree. So I'm going to draw this to a close in just 2 slides and then get on to the speakers you wanted to hear. I have 5 take-home messages just from my introduction here. The first is that the COVID-19 pandemic is neither the first nor the last that the world has or will face. We have seen that human behavior is predictable. So the way people have responded to this pandemic, the COVID-19 pandemic, was predictable based on what's happened in the past. And had we learned our lessons from the past better, we probably could have saved a lot of lives in the world. Now, I will say that I believe Canada has done a whole bunch better than other places, but from a world perspective, we could have learned from the past better. Thirdly, pandemic actions and the solutions that we take, the steps we take, need to be based on science and evidence and not on personal agendas and politics. So that was one of the lessons we did not learn. Fourthly, these pandemics still rely on public health measures and public health professionals who are key to maintaining public safety as we work towards longer term solutions to the outbreak. And that brings me to the final statement, which I hope has given some context to today's speakers and today's topic, which quite frankly is, hooray for modern science and medicine. COVID-19 is likely to be the first big pandemic that is stopped by a medical intervention. We can go back thousands of years in history, and pandemics have run their course and killed the people they want before we have come up with solutions for them that really made a difference. So this hopefully is going to be the very first major pandemic that is stopped, and it's going to be stopped, if it is, by the vaccine. So with that, I'll go on by going to the presenters that you guys have come to hear. They're going to talk about Saskatchewan's race against COVID and the things that have been done here and the things that will be done here to help ensure that we're able to beat this pandemic. So it gives me great pleasure to introduce our first 2 speakers. They are the high power leaders of VIDO Intervac here at the university. They are Dr. Volker Gertz, who is the director and CEO at VIDO, and Dr. Trina Racine, who is the associate director of vaccine development at VIDO. I'm going to invite them to turn on their videos and their microphones, and in just a second, I will turn the floor over to them. But just before you good folks start talking, I will remind the audience that we are hoping to get a number of questions for our speakers, and that to send those questions, please use the chat function at the bottom of your Zoom window. And with that, Volker and Trina, the floor is yours.
Well, thank you very much, Chancellor McCreath and Dean Rothwell for the kind introduction. So what we actually going to do is we'll share this presentation. So I'm going to start, and then as you indicated, we have Dr. Racine, our Associate Director for Vaccine Development, speak about the vaccine and the clinical trial, and then I'll come back at the end with a few summary slides. All right, so thank you very much for letting us talk about the research that's happening here at VIDO at the University of Saskatchewan. So for those of you who may not be familiar with the organization, we are a research center here at the University of Saskatchewan, off the university. We have more than 45 years now of expertise in human and animal health research. We have about 150 staff members, in fact, more like 160 now. And most importantly, in the context of this talk today, we operate some of the most advanced infrastructure in Canada and in fact around the world. And so this is just a little infographic on the organization here, but you can see we have scientists from all over the world. We are ISO certified. We operate more than $200 million worth in infrastructure. And as you can see, over the years have commercialized a number of vaccines, of which 6 were world firsts. And as it's shown here, we do a lot of infectious disease and vaccine studies per year. We have an international board of directors, which is presented here. Many of these members are actually representing our key stakeholder industries. So whether it's the livestock industries or the human health industries, but we have already about 30% international members and are bringing more international members now as we continue to grow. This is really, I think, where this talk really starts, and that is with this facility here. This is our, what we call the International Vaccine Center. This is Canada's largest high containment laboratory, and it allows us to work with these potentially dangerous pathogens like COVID-19. So there is 4 categories to pathogens, if you want. The level 1 to 2 is what we encounter in our daycares and schools. The level 3 ones are the ones that are potentially very dangerous to the individual that either gets them or works with them. And then the level 4s, like the Ebola viruses, they are the ones that also have not only a significant impact on the individual, but also on the public. And so this is a level 3 facility. We're not working with Ebola. We're not allowed to work with Ebola in this facility. This is a level 3 facility, but it is one of the largest around the world. And what it really does, it allows us to work with a number of different animal species. And you can see on the right side there, we can house in the facility and have housed anything from bats to bison. And in fact, if you would look right now, who we have in this facility, there would be a group of bison that are in these rooms right now, working on a study to look at bovine tuberculosis in these animals. And a few months ago, before COVID started, we were doing work on MERS coronavirus, so the closest cousin, and you could see on the right that we in fact were working with bats at the time, trying to understand the role of bats in the disease pathogenesis and transmission of MERS, another coronavirus. But when COVID-19 hit this last year, we were really able to quickly ramp up our work and become one of Canada's go-to places for COVID-19 research. And VIDO was the first in Canada to isolate the virus. This was done in collaboration with our colleagues at Sunnybrook and the National Microbiology Lab in Winnipeg. A sample was obtained, was sent to us. We isolated the virus and then in fact provided the virus back to these organizations who then have shared it with all the public health labs around the country. We were then the first in Canada to have an animal model established, which allows us to test vaccines and antivirals and so on. And as was mentioned in the introduction, we're now the first university with a vaccine candidate in already clinical development. VIDO scientists are members of World Health Organization expert groups, but probably most important of all of this is currently in terms of energy and time. We have been now over the last 10 months working with more than 80 different companies from around the world, or large organizations from around the world. About half of them are Canadian and the other half are international, working on finding solutions for this virus. Not only vaccines, but also looking at antivirals, at drugs, at therapeutics, trying to identify solutions on how we can control this disease. There was funding that was announced last year and was received early on in the pandemic, both from the federal government, but also very important to mention from the provincial government, who really came through and said, here is an additional support of all the research you're doing, both for the manufacturing facility as well as the research. And so there, as you can see here, there's money for the vaccine development, and you will hear about it. There is money to enhance our research capacity for all the contract research that's going on right now. And there is also money to build a vaccine manufacturing facility. And you might have heard about in the news, VIDO is currently constructing a GMP in-house manufacturing facility that will allow us in the future to make both human and animal vaccines. We were able to quickly put together a very large team early throughout the pandemic, and you can see, and I just want to acknowledge Daryl Falzerano, who you can see here on the top left. He's the coronavirus researcher who very early on started most of this work, and then we brought in more and more researchers and more collaborations and so on. And now we have, as you can see, a very large team of very, very dedicated individuals who are willing to work every day on this, long days, every weekend and so on, to help finding a solution for COVID-19. However, there's also lots of collaborators at the university on campus here who have expressed an interest and have really stepped forward and come through with working with us very closely together and working on their ideas. And you can see these collaborators are both at the Vet College as well as the College of Medicine and in the College of Dentistry. And you can see on the right side, we were also able to second researchers from other universities in Canada, from Dalhousie and the University of Manitoba, to come to VIDO to spend a year here and help us in dealing with this pandemic. We also then helped in another way dealing with this community, and this is showing here on this slide. Very early in the process, we worked closely with the SHA on identifying ways of how we can use our N95 masks that are being worn in operating rooms, and how we can decontaminate them, how we can sterilize them again so that they can be used again. And this is a process that not only we use for equipment, so you can see here's our regular rooms, and we're using a process that's called vaporous hydrogen peroxide. And it's a gas that we're using to decontaminating or sterilize equipment that is within these rooms before we would take it out of a room, or at the end of a study when we want to clean this room, we would first kill all the pathogens in there. And so we're using VHP for that process. And we were one of the first in the country in North America, in fact, to establish this process and to demonstrate that we can reuse these masks after they have gone through the decontamination process. And you can see here on the left side, there is our students volunteering for this, in fact, and now volunteering several afternoons per week when they go in and they take all these masks and they're hanging them, and they all have to be individually hung so they don't touch each other. It only works if they don't touch each other. You have to have access to the surface. They're hanging these masks, and then we treat them, and then they collect them all, and they're being shipped back to the SHA. So another way of showing how we have been able to use this facility and our research expertise here to reach out and help our communities. But this is now really talking about the vaccine that we're making and where we are with this. And I'll provide a little bit of an introduction, and Trina will tell you exactly where we are in terms of clinical trials. So we all have seen, in fact, we just in Doug's slides saw a picture of the virus. So here's a very rough schematic of the virus, how it looks. And all I want to point out is that most vaccines that are in development right now are focusing on the structure of the surface here, this yellow structure, which of course in real life is not yellow, but here for simplicity is colored in yellow. And so you can see this structure here, this spike protein, we call it, that's being used by the virus to attach to the host cell. And so it binds to a receptor, and when it does, the virus then gets access to the inside of the cell. It can enter the cell and then start causing disease. And so that's why most vaccines these days are focusing on preventing the attachment of the virus to the host cell. And so they're targeting the spike protein, and that's why ours and many others are now focusing on the spike vaccine as the vaccine antigen, if you want, as the material that we're injecting. So this is just shown in real life here. You can see here's a virus, which is in this case labeled, and there's a green fluorescent protein involved there. But what this graph is showing is how these viral particles are trying to get into the cell, which is shown here. So this is the cell body, and these viruses are trying to get in. And schematically, it looks like this. There is your spike protein, the structure on the viral surface that now binds to the receptor that is found on the surface of the cell. And then the virus can get in and causes the damage to the cell and making lots and lots of new copies that are then being shed. So what these vaccines, all of the vaccines are really aiming for is two things. Number one, prevent the attachment of the virus to the cell. And we do this through the development of antibodies that can block the binding to the receptor and therefore neutralize the virus. And that's why we call these neutralizing antibodies. And they're very, very important in protecting us from or against this disease. And then we are looking for T cells that can come in, and these are specific immune cells that we have, and they can kill other cells, and they predominantly kill cells that are infected. And so in this case, they would be recognizing that this cell is infected with the virus. And so the strategy the immune system uses there is to kill, to destroy the cell, and therefore also destroy the virus with it. And so most vaccines now are aiming for high levels of antibodies, but also high levels or good stimulation of T cells. And so this is shown here. This is the airway in your nasal surfaces. And so the virus would be on the outside when you breathe it in, and now it binds to these receptors on the surface and finds its way in. And that's then when we see these problems. So where we want these immune cells to be is essentially up here, the antibodies to prevent the binding of the virus to the cell, and then T cells right here in the tissue that it potentially could infect these, kill these infected cells. And so different vaccine strategies all are aiming now at the COVID-19, at the spike protein on the COVID-19 virus. So you can either have a whole life attenuated virus. There is not many vaccines that are currently based on that. There's a few in China that are following that procedure. Most vaccines are focusing on the actual structure itself, so the spike protein. And so the first one that is relevant here is the DNA or the RNA vaccines as they're now approved. So the Pfizer, Moderna vaccines, they are using a piece of the genetic code, a partial fragment here that specifically encodes for the structure on the surface. So all we're injecting here is the genetic information for the cell to then make a structure like this, and then for the immune system to recognize it and make an immune response to. Or you can just deliver it with the vaccine. So instead of providing the genetic code, the instruction manual of how to make it, we are providing the protein itself. And that is what the VIDO vaccine is doing. And that is, for example, also what the Novavax vaccine is doing. And then you can also deliver this, the genetic information and the protein, with another vector, another harmless virus, an adenovirus. And that is what, for example, the Oxford vaccine, the AstraZeneca vaccine, is doing. But all of them are aiming to induce an immune response against the spike protein, which is this part on the structure of the virus here, allowing the virus to attach. And so the goal is that if we are getting antibody responses or even T cell responses that block the binding of the virus to the receptor, it can no longer enter the cell and therefore could no longer cause disease. And so this is now where I'm letting Trina take this over, and so she can talk to you about the stages in vaccine development and where we are with our own vaccine.
All right, thank you so much, Dr. Gertz. So as he mentioned, I'm gonna now talk about the first stages of what goes into getting a vaccine into the human population or even into the livestock population. And so the first thing we do, of course, is some exploratory analysis. We got to figure out what's the pathogen that we're interested in and how do we think we can prevent it from infecting. And once we kind of come up with that initial vaccine design, we have to test it in what we call preclinical testing. And so this typically takes place first in small animals, and then we work our way up. And once that data is generated and it's encouraging, we are able then to submit that information to regulatory authorities like Health Canada. And that's called a clinical trial application. And in that clinical trial application, we're going to provide the regulatory agency with information on the effectiveness of our vaccine in those preclinical studies, but also information on how we manufacture the vaccine to demonstrate that the product that we've made is it's safe, it's pure, that there are no contaminants, it's not, that it's sterile, and that it can be kept in the vial that it's stored in for long periods of time without having to worry about the product degrading. And so the very first stage of clinical development is called a phase 1 trial. And phase 1 trials typically occur in healthy individuals and normally in a group of less than 100 people. And this first phase of clinical testing is really to look at the safety of the vaccine. And once we have a good safety signal for the vaccine, we then move on into phase 2 clinical trials. And now we're looking at hundreds of people, potentially even a few thousand people. And now we're still examining the safety of the vaccine, but we're also wanting to look at the immune response that the vaccine can generate. Are we producing antibodies? Are we producing a T-cell response? And then finally, we're going to move into a phase 3 trial. Now, this is an efficacy trial. These are the large tens of thousands of people trial, usually happening at multiple sites around the world. And for a phase 3 trial to truly be effective in determining efficacy, we need to have it tested where there's actually active circulation of the virus, the pathogen of interest. So that we can see whether the people who received the vaccine are protected compared to those that did not receive the vaccine. And we should see more infections in that other group that didn't receive the vaccine. Now, once we have that efficacy data, organizations, companies that are sponsoring those clinical trials will then bring that information to Health Canada, here in Canada, or other regulatory agencies for review, because they're going to look at all of the data that's been accumulated up until that point, all that preclinical data. They're going to look at it all again. They're going to look at all the clinical data that's been generated. And again, also look at the method of manufacture of the vaccine to make sure that it'll be safe and that we can do it consistently. You can produce multiple lots, multiple batches of the vaccine, and we can do it consistently. And only then would they allow it to be distributed to the public. And so we in Canada and at VIDO, we are currently, as Dr. Gertz indicated earlier, in phase 1 testing of the vaccine that we've generated at VIDO. So this is just a quick kind of timeline to show you what we've done and how long it's taken us to do it. And so for us at VIDO, the design of our vaccine started off as soon as there was a sequence available to the public that was published January of last year in 2020. And due to the experience that we had in-house working on a MERS coronavirus vaccine, we were able to generate our own lab scale batches of potential vaccine candidates in January and February of last year, and we were able to test them in our level 3 facility in proof of concept studies between March and June. And back in March, we started the GMP manufacturing process for our vaccine candidates. And then one of the steps that we need to take before we can actually commence clinical trials is to do what we call preclinical toxicology studies. And this, again, is just another way of demonstrating the safety of the vaccine before it goes into humans. And so we submitted our clinical trial application for our first phase 1 trial in December of last year and received that approval from Health Canada. And we started our clinical trials recruiting in January, and our first doses were just provided. So that's been very exciting for us. Throughout this time, we've done a number of different presentations to various organizations and committees within the country and funding organizations, et cetera, to just kind of demonstrate the data and the work that we've done so far, one, to raise awareness of the vaccines that we're developing, but also to help receive some funds. And so to talk about the 2 vaccines that we are developing here. We have named them COVAC-1 and COVAC-2, and both of these vaccines are using the same S1 antigen. So we are using a recombinant protein subunit-based vaccine, and that was one of the kind of platforms that Dr. Gertz talked about earlier. And so what we're doing is we've taken the S1 portion of that whole spike protein, so just that little area that's indicated up on the top of the slide here, and we're combining that S1 antigen with an adjuvant. An adjuvant is a compound that just helps boost the immune system to respond to the protein or antigen of interest. And so our COVAC-1 vaccine actually is composed of, or is paired with a triple adjuvant that has been developed here at VIDO over the last decade and has shown to produce a very strong T-cell response, which we are confident will help produce a very broad immunity to our vaccine. And the COVAC-2 vaccine formulation is formulated then with an adjuvant called SWE, which is a squalene-based or oil and water-based adjuvant. And it too should give a good, nice Th1 and Th2 response. So now that we had our vaccine, we have to test it, of course. And as Dr. Gertz mentioned earlier, we were one of the first organizations in the country to develop animal models against SARS-CoV-2. And the first model that we developed was a ferret model, and then we subsequently followed that up with a hamster model. And so both of these models are based on the fact that the ACE2 receptor that the virus binds to in these animals is similar to the ones that we have in humans. And so the virus is able to actually bind to that receptor in the animals and infect them. Now, in ferrets, ferret is a very mild clinical disease, but they do get infected in their upper respiratory tract, whereas hamsters develop a little bit more of a severe disease. So this is just some data to demonstrate to you that our vaccine candidates do in fact produce neutralizing antibodies in our animal models. And so the day minus 70 and day minus 42 are in relation to when the animals were challenged with the virus, when they actually received virus. And so we vaccinated animals at day minus 70, at day minus 42, and then at day 0, we challenged them with the virus. And we can see that prior to challenge, both of our vaccine candidates are producing significant neutralizing antibodies compared to the mock vaccinated animals. And that 14 days into the challenge, we can see that our neutralizing antibody response has increased even more, and that while the mock infected animals do have a response, that's what the response is due to a natural infection. And you can see that the level of antibodies at day 0 in our vaccinated animals are quite similar to what the natural infection response would be. And this is just one last piece of data to demonstrate how our vaccine is protecting against clinical signs of disease. And so the pink triangles or purple triangles are animals that were not vaccinated or mock vaccinated and then challenged with the virus. And we can see that they lose about 15% of their body weight at day 6 post-infection. But our different vaccine candidates, we can prevent that loss of weight loss. So that is protecting the animal against disease. So that's what we're looking for in a vaccine candidate. So this is just another kind of timeline to show you the work that we've done over this past year and the different steps that we're taking and are still taking to get the product through clinical testing and hopefully into licensure. And just to point out, the phase 1/2 clinical trial has now started, and we hope to commence phase 3 testing later this year. And this is the safety toxicology study that we conducted in rabbits. This was done by a third-party organization that conducts experiments under what we call good laboratory practices. This is just a kind of a step up on how you conduct an experiment when it comes to documentation and reporting, so that we know that the study was conducted really well and efficiently, and anybody could reproduce that study. And so this toxicology study, we ran it for both of our vaccine candidates, and neither one showed any kind of mortalities or clinical signs in the animals. And there was no gross or microscopic pathology, which means that the vaccine itself was not harmful to the animals. And also, really importantly, is that in both studies, both vaccine candidates produced strong antibody responses. And so now to our clinical study design. we are currently conducting a phase 1/2 clinical trial of our COVAX vaccines in healthy adults. The study is sponsored by the University of Saskatchewan and is being conducted at the Canadian Centre for Vaccinology in Halifax at the part of the Dalhousie University and IWK Health Centre. So COVAX 2 is the first vaccine formulation that we have gotten into the clinic, and our COVAX 1 vaccine, we anticipate being able to bring into the clinic by this spring, around May of this year. And so our study is a randomized, placebo-controlled, observer-blind dose escalation study. So what this means is that both the study staff that are going to be interacting with the study volunteers and the volunteer themselves don't know whether they received the vaccine or a placebo. And we're doing a dose escalation study to see what dose is necessary to get a good response in individuals. So we have 3 dose levels and also 3 age categories. We're starting in individuals 18 to 54, and then we're going to move up into individuals 55 to 69, and then 70 and above. And that's just to kind of see whether our vaccine is able to produce a strong immune response in all the age categories. And we are going to be recruiting 108 volunteers per vaccine formulation. And we are already underway. We're already thinking about the phase 3 studies, what's the design going to look like. And we're also have started producing GMP material for that trial. So that was the clinical trial and the vaccine. But now, as Dr. Gertz alluded to earlier, we are also building a vaccine manufacturing facility here at VIDO. And so that facility is going to be housed within our Intervac building, so that the red circle area that Dr. Gertz is showing there. And so this vaccine facility is going to be what we call a pilot scale GMP facility. It's going to be a very unique facility in that we are building it to be very flexible, in that we are going to be able to develop vaccines against veterinary pathogens as well as human pathogens. We are not limiting ourselves to one vaccine platform type. We're going to be able to develop vaccines for live attenuated, inactivated, subunit-based vaccines, viral vectored vaccines. And so that way we have more opportunity to develop products that could eventually reach the market. And we estimate, depending on the actual manufacturing process and how good of a production runs we can get and the strength of the dose that we need to provide, we could produce potentially up to 20 to 40 million doses per year in our facility. And the construction is already ongoing. Equipment is being purchased, and we anticipate that construction will be completed by the fall of this year, and we will be conducting initial production runs early in 2022. Back to you, Dr. Gertz.
Okay, so I'm going to jump back in here then. We wanted to also take an opportunity just talk a little bit about what can we learn from this. And Dr. Brothwell had a very interesting presentation alluding to an earlier pandemic there, and you probably saw all these similarities that he purposely put in there. This is really, not sure if it's going to happen exactly in this way, but it's for sure that there will be another emerging disease outbreak, whether it's in animals or humans, in the near future. And so if you look back over the last 10 years, we had SARS-1, or 15 years, we had SARS-1, we had influenza, we had MERS, we had Zika, now we have SARS-2. In animals, we're also seeing these emerging diseases. And there is this news clip on the right side of a disease in pigs. It's called African swine fever, which is a disease of pigs only. It's not affecting humans, but it is a huge threat to the Canadian pork industry. It's currently circulating in Asia and China and other countries. It's not really an emerging disease in that we actually know this disease for quite a while. But what we have found now over the last few years, that this disease has made its way now into Germany, where it is now in wild boar, and Germany is the largest pork producer in Europe. And so we all think that it's actually not a question if this disease will make it here, it's more a question of when will it make it here. And we currently don't have vaccines for this disease available. The economic impact to Canada will be that Canada will lose its ability to trade its pork products with other countries. And the Canadian pork industry is an export industry. More than 60% of our pork products are going to other countries. And so having other countries having or closing their borders for our products is going to be a huge problem economically. And so the point of this is that these emerging diseases will continue to occur. Scientifically, we're expecting every year about three new diseases emerging, and every third to fourth year there's a larger event. And so what we need to really think about for the country to be better prepared is to have these national centers that are focused on these emerging diseases and then have the ability to rapidly respond. And the only way they can do it is essentially, or in our mind at least, by having vertically integrated three main components, as you see here in blue. The ability to work with these pathogens in high containment, to have an in-house manufacturing facility that allows you to quickly make clinical grade material, and then have the ability to work with a wide range of animals from which these diseases emerge. And so this is where we think VIDO can help the country to be better prepared for the future. We're currently operating, as we mentioned, Canada's largest high containment laboratory. We're building a manufacturing facility in-house already, and we have the expertise of working with both human and animal pathogens. And so our research right now is really focusing on this. Why do we have to wait until a new disease emerges, which normally then takes 10 to 15 years before we can come up with a vaccine? Now we're seeing with COVID that within 1 or 2 years we might be able to do it. But even that still accounts for millions of lives lost and trillions of dollars in economic losses. So the question is, why do we have to wait? Can we not predict what the next pathogen might look like and make a vaccine that is available, stockpiled before such disease even breaks out? And you might say that's all blue sky, that's dreaming, but I would argue with you that the science is there to now, by using a combination of structural biology and bioinformatics, artificial intelligence, and so on, and then facilities like ours that have the ability to work with all these different animals, we might be able to predict what the next pathogen might be and have vaccines that could be stockpiled and ready to go. So in this case of COVID-19, it would be a plane leaving the WHO in Geneva, flying to Wuhan and handing out 10 million of vaccine doses early on in the outbreak. And even if these vaccines are not 95% effective, maybe only 80 to 85% effective, they would have helped in controlling the initial outbreak. And so this is much of what VIDO is going to focus on in the future, trying to come up with both human and animal vaccines that can be used before a disease breaks out. And so that's wrapping it up. Just to acknowledge the great support we have received over the last year and a bit, both from the federal as well as the provincial government, and then of course CHR and many private funders who actually have come forward now and are supporting what we're doing here at VIDO and Tobacco. So with that, we're going back to I guess the discussions, and Doug, back to you.
Thank you very much, Volker. Greatly appreciated. What a wonderful duo of presentations, and I'm happy to let you know that you kept the discussion at a level that even a public health dentist could understand it. So thank you very much for doing that. We have received some questions through the chat. I believe we would have time for more, so if anyone has additional questions, please feel free to submit those. But let's start out with the first question, and Dr. Racine and Dr. Gertz, you guys can decide who answers the question between the two of you. I will just bring the question forward. So the first one is the spike protein fragment that your vaccine targets conserved between different SARS-CoV variants?
So of course, that's something we're looking at right now. So we're addressing that right now and making even slight adjustments to the antigen to have this vaccine be more effective against as many variants as possible.
Excellent. And of course, I guess the idea is, the worry is that we'll get vaccinated against one set of a vaccine that works on one set of viruses, and then there'll be a vaccine in the future, a variant in the future that would require a different vaccine. So I'll just add my own question on top of that one then. What do you think is the chance that we're going to be going back for annual COVID shots the way that we go back for annual flu shots?
I think that chance is very high. I'll let Trina comment on that too, but I think that's something that I would think is most likely that there will be maybe annual boosts or maybe a little bit longer than a year, every 2 years or so on. And I think we will also see that some of these vaccines will adjust over the next few years. The other component that I didn't mention in my previous response is this adjuvant that Trina was introducing. By getting a very broad immune response, there is a lot of evidence that you then also are more effective against these variants. And so I think that's another thing we will see in the second generation of vaccines coming forward, that people will not only adjust the antigen they're using, but they will also adjust the adjuvant and therefore broaden the immune response. Trina?
Yeah, so I concur with everything you've just said. I do agree that we'll probably have to come back for multiple doses, whether they be, like Dr. Gertz said, every year or every other year. It will be dependent on the breadth of immunity that the vaccine provides and how quickly the virus mutates. Influenza mutates quite rapidly. And so that's one of the reasons why we have to get that new flu shot every year, is because the virus that's circulating is different than what was last year. And so the last year's dose probably wouldn't protect you this year. And so that will all depend on what the virus does. And we do feel that the adjuvants that we are using will help provide us a larger breadth of immunity than some of the other vaccine platforms that are currently out there.
Thank you. I'll go on to the second question then. We have a submitted question. Do you plan to have any trials in children? And if so, why? And if not, why not?
I'll let Trina comment on that first.
So we don't have any trials planned in children just yet, but we are in the planning stages. We're looking at what that would entail, what age groups we would start with. We'd most likely start with, your 13 to 17-year-olds and work down, just as we worked from, adults worked up to more older individuals. But that will be dependent on funding. So we will see what we can do.
Thank you, Volker.
Oh, I don't really have anything to add there. It's a good question, though, and that's something that for all vaccines will have to be done, to take it into children. Also, the question of pregnant mothers, for example, there's a lot of questions that clinically will have to be addressed, and they're currently being discussed by the WHO and other international organizations.
Thank you. We'll go on to our third audience question then. How is this virus different than SARS-1? This person says, I believe there was a vaccine developed, but the virus did not spread to the same degree as SARS-2, COVID. And did the collective not learn from the SARS-1 epidemic vaccine? And why were we not able to carry that forward?
So I think my answer to that would be that we actually learned a lot from SARS-1 and also from MERS. So we learned a lot about these coronaviruses, and in fact also by working on animal coronaviruses, the ability to quickly identify the spike protein as the target for this vaccine and really understanding that neutralizing antibodies are so critical has really enabled us to develop a vaccine in a year. Without that information, nobody would have been able to make a vaccine in a year. So it's really all the background from SARS-1 and other coronaviruses that has helped us to make this more rapid or faster. The other thing I think we learned from this is, and maybe we've forgotten a little bit, and that's more on the political level, that SARS-1 indicated, and then of course many other events since then, the impact of these emerging diseases. And at one point we had pandemic preparedness programs in place and so on, and maybe because of a change in priorities over the last decade or so, some of these are not at the stage where they maybe should have been. And so this is, I think, a big take-home message from this pandemic, that hopefully we will not forget in the future, and we will put these programs in place that will then allow us to more effectively respond to the next event.
Thank you. Dr. Racine, anything to add to that?
No, I think that was very good. Very nice response.
Okay. I'll go on then to the next question. Is government approval to conduct vaccine trials a major factor in the rapid development of vaccines, or is the funding the more relevant factor in there?
I'm going to take that one to start off with. So they're both important factors. Without any funding, there's no research to even get to the stage where the regulatory approval is a concern, right? So the funding needs to be in place even before there's a problem, right? So that leads back to the previous question of, what happened with SARS-1. And I have to say that I'm very happy with Health Canada's response to regulatory submissions this past year. They've been very quick. They've modified their normal timelines, and so they're doing what they can to help us move as quickly as possible, but not jeopardizing the safety of the public. They're still doing their full evaluation, still looking at absolutely everything, but they're just working so much harder, so much longer to provide that in a much faster time frame.
Thank you for that context. It's good to know that we seem to be doing quite well in Canada, so that is quite reassuring to hear that from you. I think we have time for one, possibly two more questions. So let me find it here. So your presentation talked about weight loss as in the, I think they were animal models, but in the subjects that was, and the vaccine seemed to protect against it. So the question is protecting from weight loss a different outcome from getting full immunity? And does this mean that the vaccine might help prevent the serious outcomes, even if you're still able to, to contract the disease, but just not have the serious outcome? So I don't know if those two tie together, but we have a question about it.
Dr. VIDO, you want to address that? Was it your slide?
Okay, sure. So the weight loss is an animal model kind of clinical sign, so that not all animals, ferrets tend not to lose as much weight when infected, but hamsters do. So it's just one of, like I say, the outputs of infection and a clinical sign in that model. But yes, it's how we kind of look at how effective the vaccine will be in humans. And so if you can protect against weight loss, it's kind of it suggests that we're going to be able to protect against those severe clinical signs in humans. Dr.
Thank you very much. Dr. Gertz, anything to add to that? Oh, good. I'll go on then to what probably will be our last question then. Someone has asked, what are the side effects of the vaccine?
So it's a subunit-based vaccine. So this is a platform and a technology that is already in licensed vaccines. Many of our childhood-based vaccines are subunit-based vaccines. So there are currently known severe side effects. I mean, they're very rare occasions of things happening, but your traditional, muscle ache at the injection site and, a little bit of redness and swelling, those kind of things. But the side effects are the reason why we do these initial safety trials in a small number of people to see if there's anything significant that pops up right away, and why you expand on the number of people as you are testing the vaccine.
Thank you very much for that. Nice quick answer. And so I think we have time for one last question. This will end up being the last one. The question is, if you get a vaccination, say from Pfizer, and a different vaccine is developed later in the year, would it be recommended to go and receive the new vaccine as well?
Yes. So the answer is yes. The vaccine that we're working on, as Trina indicated, is a well-proven technology. It's actually a good technology to be a booster vaccine. So even if you had received an RNA vaccine before, it would make a lot of sense to boost with our vaccine in the future.
Excellent. Thank you very much, speakers. Greatly appreciated, Dr. Gertz and Dr. Racine. With that, that comes to the end of our 20-minute question period, and I'm going to move on now and introduce our third speaker, who will move the discussion into the direction of the mechanisms and logistics of developing the vaccine in Saskatchewan. So it is my pleasure to introduce Dr. Corey Neudorf, who is a consulting medical health officer to the Saskatchewan Health Authority, and who in a recent personal Zoom call, I found out that the two of us were within just a couple of years of each other of studying public health at the University of Toronto. And so I feel now like Corey's almost a classmate of mine, not quite, but almost. And so with that introduction, Corey, I will turn the floor over to you. Dr. Neudorf, the floor is yours.
Thank you. So I've been asked to speak about the vaccine administration, and in order to do that, I want to speak a little bit about the background of where we've gotten to and control of COVID overall as we start looking at where vaccines fit in the model of control. First, we'll just talk about the background. Obviously, we've heard about this already before with the previous talks, but it's hard to believe that really it's just a little over a year ago where we had this identification of this novel infection and how quickly it spread globally. But our first cases in Canada early in 2020 really resulted in some pretty substantial initial control measures put in place. Given the fact that there's a lot of unknowns, they had to be quite broad-based, and it was in keeping with what many other countries were doing, as we know, and in response to reports of how severe the impact had been in some of the earlier places hit. In addition to that, there was a lot of work that was going on by researchers using computer models now to look at learning from what was known about this pandemic, previous pandemics. And as we saw how things were unfolding in other parts of the world, we could gradually refine models to see just what was the potential for the spread of this disease, knowing the likelihood, the seriousness of the virus, how easy it is to spread from one person to another. And as we were starting to get to understand the role of touch in spread versus droplets versus smaller droplets or potential for aerosol. So these models gradually became tighter and tighter. But we did start to understand also what was effective and what types of interventions then would work against that type of spread. So modelers also built in those provisos as well and showed what kind of trajectories, how you could impact the trajectory. And I'll show you some examples of that. And we got introduced to this concept of the hammer and the dance, which is this idea that you hit the virus really hard to begin with really severe restrictions. You gradually start letting those restrictions off. And then as you see cases start to increase again, you might have to then get more strict again. And you see this, this dance, as you will, of cases starting to come back and then being restrained again. We also started to see inequities. We knew that this always happens in pandemics, but given the way this virus spreads, we knew that there would be inequities in both infections as well as complications of the virus, but also unintended consequences because the restrictions, of course, were so severe. But the need was there to act very quickly and then put mitigating measures in place to try to reduce some of those negative impacts. This is just a graphic from the Chief Public Health Officer of Canada's report from October 2020 that I was consulting on, basically just showing this two-pronged approach here and looking at how things like our social determinants of health and structural determinants impact both on our susceptibility to the virus, given where people live and work, overcrowding situations, for example, the impact of various types of jobs that have different potential then for exposure to the public. Essentially, it became very obvious early on that an all-of-government approach was going to be needed to respond to control, and that we would see in parallel health systems activating incident command centers to be able to handle simultaneously this massive contact tracing and testing protocol, along with getting the system ready for potential influx of cases as cases starting to materialize. And of course, in Saskatchewan, we had the added complication of being fairly early in our health system transformation. So many of the structures that you would hope to be in place were still being put in place. This allowed us in some ways to respond faster, given the fact that many decisions were centralized. But on the other side, some issues were harder because we were caught midstream with new management structures that hadn't been fully brought up to speed, say, with a single pandemic plan for the province. This is another graphic from that same report showing that there was a need to take an equity approach to managing COVID. And we saw that because of this, the way the virus spreads and the way it impacts on people, I've heard it said the pandemic exposed cracks in our social safety nets, and it also amplified pre-existing health inequities in societies around the world, including Canada. So you had this phenomenon of disproportionate rates of COVID cases, hospitalizations, ICU admissions, and deaths in certain vulnerable populations. This included, obviously, increasing severity with how old people were, but we also saw increased cases, hospitalizations, and deaths in those populations living in overcrowded situations or with underlying medical conditions, or even in racialized groups or those living in poverty. And of course, those who are considered essential service providers were disproportionately in those groups and therefore exposed to the virus more, perhaps not having a choice to stay at home or not having their particular work closed, and therefore having to ride crowded transit or ride sharing, working often, especially early on, without adequate personal protective equipment. And of course, coming back then to multi-generational homes or crowded conditions, so that if they did get the disease or need to self-isolate, that became more difficult. So this pandemic response, of course, needs to be nuanced, and different types of communication, interventions, and protocols put in place to mitigate some of those impacts. So, of course, like many others, the University of Saskatchewan researchers, a lot of us had to pivot our work into COVID research in 2020, and there could be pages and pages listed here. I've just listed a few that I'm involved with, looking at how is it that the public health capacity and our governance models in place in Canada are allowing us to respond better or worse to COVID than other places. We've got a social contour study going on with Spiru, looking at trajectories of adherence to public health measures and people's intention to be immunized that's being tracked. There's seroprevalence studies looking at what is the rate of disease that's been found in the public overall, not just in those tested because of symptoms, but in the overall population. Epidemiologic studies and computer modeling, which I'll be showing some results of in just a minute. And of course, vaccine acceptance studies are being looked at now, including a proposal to say just how might we get certain subgroups to make sure they see the importance of getting immunized, such as our own students here at University of Saskatchewan. And then above all of that, a lot of researchers were taking part in evidence support teams to look at this rapidly emerging evidence and how do you translate it for policymakers so that they can make use of the best evidence in making their decisions. So this is just an example of this hammer and the dance idea I was talking about before. This idea that Depending on the type of restrictions that we put in place and how much people adhere to those, we might see these successive waves take different shapes with small initial waves with larger subsequent waves, or could we get into this wandering baseline and sort of more of a low level wavering as you see toward the bottom. So these were some of the modeling scenarios that came forward back in November, if you remember, from our modelers giving to provincial governments, saying that in our current trajectory, we weren't seeing very high levels of mask adoption or following of some of the other restrictions on social gatherings. And there was an optimistic scenario in green there that was listed. If we could get higher levels of mask wearing and get people to reduce their social gatherings, next slide, then what we would see is reductions in numbers of cases per day, reductions in numbers of hospitalizations and deaths per day, and that bar that's sort of in the green, third column over from the left, is showing the optimistic scenario, and you'll recognize in those numbers that that's fewer than what we have right now. The status quo at that time, if partial control measures were put in place, were the next row over, and you'll see we're not quite at that level yet. So we're somewhere in between right now, and that's largely because the restrictions that have been put in place and people's adherence to them are somewhere in between the optimistic and the status quo scenario we saw in November. So this is just an example of modeling where it was showing here that if we could put in place really strict restrictions back in November, we could see a very sharp movement down in green to that curve, and then a slow decline into December, January, February. And if we didn't do much, we would see this exponential rise just at different rates. So this is the actual scenario now. This is Canada's curve, and we're looking at this first wave that we had in the spring, a little bit of a dance going on there in the summertime, and then this second wave that started in the fall. And then Canada-wide, we're starting to see a little bit of a tail off of that second wave right now as we get into January, early February. This is the same data, but now just showing the two waves of hospitalizations and ICU patients and patients on ventilators. So our current status in Canada is showing, unfortunately, that Saskatchewan has the highest per capita case rate, and it's been sustained at relatively high levels now for a number of months. And that is a result of decisions around not going into as much restriction as we've seen in places like Ontario, Manitoba, or even Alberta more recently, but maintaining more businesses open and not having quite as many restrictions. So we are seeing a higher case rate in our province than neighboring provinces. And unfortunately, that means with deaths as well, because what we see with deaths is about 2 to 6 weeks out, we see the hospitalizations and deaths start increasing as you see cases increase. So the deaths that we're experiencing now are a result of cases that occurred a month or a month and a half ago. And initially in our pandemic, unlike some of the other provinces, we were able to keep it out of long-term care facilities. So our death rates were much, much lower than anywhere else in Canada. But in the second wave, especially the last few months, we've seen more outbreaks in long-term care, and those death rates have certainly come up a lot higher than even what those optimistic scenarios were back in the November modeling. That just basically shows that it's impossible to circle a fence, if you will, around long-term care, even with restrictions and visitation. People do, even those who work there can come in and with asymptomatic infections, not even realize that spread is starting to occur. This is just another bit of epidemiology, more at a geographic level, and it's just meant to show that you have these waves or transitions, if you will, of who the outbreak is affecting different parts of the province. started out in travelers and those returning from vacations down south. Then it moved through different phases in the spring and summer through different work situations, has various restrictions put in place. Eventually, the main driver has been community spread, and it's pretty much province-wide. This is just to show the difference in age groups. So we have this bulk of cases that are out there in the younger age groups, and the people in their 20s, 30s, 40s. But the lines showing the numbers of hospitalizations and deaths. And so you can see how the more serious conditions or result of COVID is disproportionately affecting those who are older. But of course, you also see that there's still a significant number of hospitalizations and deaths occurring in the younger ages as well. So our current situation, this is just an example of indicators that we look at to see, how are things progressing. Essentially, we still have a virus that is spreading a little bit faster than to get it under control. The R number that we've heard about, this reproductive number, is slightly above 1 as of the end of January, staying pretty stable there. That means it's still growing. The rate of growth is slowing down, but so is testing. And so if not as many people are getting tested, you don't find as many cases. So are we really seeing a true drop in cases, or is it a secondary effect because of drops in testing? A clue to that is in our percent positivity, which is still pretty high. It's come down slowly, but it's still showing that a high proportion of those people who are going for testing are positive, which means we're probably not testing enough yet. And then, of course, our deaths and hospitalizations aren't decreasing very quickly either, and the system pressure is still very high, and the number of contacts per case is still too high, which means we're getting outbreaks and more spread. Our way recently, of course, is COVID variants we've heard about, and all viruses vary, they change, they mutate over time. Some of those help them, some of them don't. And in this case, we've had some variants that seem to show some spread of some changes that result in easier spread, not as much in terms of increased severity. And so a challenge coming before us is, can we get people immunized quick enough before the new variants take over and spread becomes even more rampant? So the key takeaways from this epi is just that we currently still have the highest rate of COVID cases in the country. We have a high likelihood that community transmission will increase, that our hospitalizations and deaths will remain high, that our epidemic doubling time is still staying stable. It's not increasing, but it's not decreasing either. And that we have this problem with high test positivity and yet low testing rates. So we are still in a precarious situation in our province. You've probably seen variations of this graph all over, but essentially what it's showing is that you need multiple layers of protection in a pandemic. And so any one thing by itself is not enough. There's always a lack of 100% protection. And so by adding layers of protection with physical distancing, hand hygiene, masks, limiting crowding and increasing ventilation, et cetera, all of these things add up to layers of protection. It's important to notice that vaccines are the final layer that you see in this slide. It's not something that's meant to be the first thing you look at. And why is that? Well, because it takes time, as we just heard, to produce them and to get them into arms. And they too aren't 100% effective. And so we need these other layers of protection in place as well. So over time here, we've now seen that some countries are able to immunize their population a lot faster than others. Lots of different reasons for that. Sometimes it's because a country has been able to get a certain supply of vaccine, but they have a relatively small population, so they can immunize people a higher proportion more quickly. Some have spent more money on vaccine or been a little bit further or up the list in terms of contacting the manufacturers first. Some of them have a better health system designed to enable people to be identified and called in quicker. Lots of different reasons, but Canada is sitting not that high on the list yet in terms of getting vaccine into arms. We certainly have delivered the vaccine that we have, but the timing of the delivery will make our initial rollout slower than some countries, but catch up should be very quick as vaccines start arriving. So what makes this administration so difficult? Certainly there's lots of issues here around the vaccine itself, the methods, logistics, complexities of distribution. It all differs depending on the platform that was used. Some of these vaccines, of course, need lower temperatures to be stored at, super cold storage, and some are more volatile in terms of if you shake them up, they aren't as useful. So you have to be careful in handling. You need different kinds of training. There's procurement challenges, logistic challenges, and even supply challenges in terms of getting enough syringes of the right type, as we just heard with these low volume syringes needed for the current vaccines. You have to get PPE for workers and design the actual delivery points. All kinds of issues that need to be thought of to just get ready for administration. And then the logistics by themselves, the storage and cold chain issues. You want to make sure that vaccine that's getting out there is still going to be safe and viable when it's injected into someone's arm. So you have to think about that stability of that cold chain, and that varies as well with the different vaccine technologies. There's always a need when you're doing a mass campaign for increasing the numbers and types of providers as well. So as we get more vaccines and vaccine supplies, we need to get more providers involved and train them up in delivering it. So the more people you get involved, the more complicated it is. There's the need to balance between speed and ensuring that the conditions that you're providing the vaccine don't lead to a super spreader event. So you don't want to have everything focused on mass clinics and gather a whole bunch of people together when it takes a week and a half to 2 weeks to start showing effectiveness of the vaccine and actually have outbreaks going on in the meantime. Of course, you need to maintain other vaccine programs and add this program to it. You don't want to leave us vulnerable to other vaccine preventable outbreaks while we're focused on this program. So some of these resources have to be added to existing. And then, of course, the prioritization battles that we've heard. Everybody's making a strong case as to why they should be next in line, and they're all viable reasons as well. Bottom line, as we've just heard in the last presentation, is domestic production is certainly needed if we really want to get serious about providing vaccine in a timely manner. We've had this as part of our pandemic plans for decades, but it hasn't been heeded by previous administrations in this country. So we've had federal vaccine rollout guidelines in place. They've been in place for pandemic plans for decades, and they just get dusted off and refreshed based on whatever disease it is that we're facing. So in this case, with the specific types of risks for both spread and who has the most complications, the federal government gave these kind of guidelines for who should be first in line, as you can see. And then Saskatchewan strategy. We've taken those pieces of advice from federal government and looked at, okay, what's our campaign goal? It's nuanced across the country based on how is the epidemic being experienced in that locality. And so in Saskatchewan, a decision was made to focus on minimizing severe illness and death and protecting the most vulnerable, that followed by protecting healthcare capacity and minimizing spread and protecting critical infrastructure. So if those are your goals, then obviously phase one is going to focus on the most high-risk populations. So that's people of advanced age and the healthcare workers working with them. And then as you get into phase two, looking at who's next up. So it's primarily those of the next age group and certain people with underlying medical conditions that make them more susceptible as well. So age is the primary risk factor that's been decided in our province. It also has an impact on delivering more efficiently in that manner as well. So you'll see here, this is taken from the government's website and looking at who's identified as phase 1 and then who's identified in phase 2. And this ability to kind of simultaneously look at those in their 60s and with certain underlying conditions in the first part of phase 2. This is just a little more detail about where we're at right now with phase 1 priority populations. So those who are vulnerable populations in our north, where the highest attack rates are happening right now, followed by those also with highest complication rates, like those above 80 and those in their 70s. And of course, where we started in December and early January with long-term care and the staff there, along with healthcare workers that are working with COVID patients. Important to note that even if we get all the vaccines that we were expecting in Q1, we won't have enough just to get at this first priority population group. So here's where we're at right now. I won't go into detail because of time, but you'll notice that we've hit over 100% in many of the markets here, and that's because the initial Pfizer product that said it was for 5 doses, we found that you could get 6 doses in some cases, and certainly with switching to low volume syringes, you can get that more consistently. So why focus on age? Well, this is the obvious epidemiology here. The number of deaths that we're seeing is very much age related, and the percentage of deaths as well, as you look at that, very, very strongly age related. So if your focus is to decrease those serious impacts first, when there's limited supply, it makes a lot of sense to focus on age. And then once you get those groups immunized, then look at the next level, which is where are the outbreaks or more concentrated populations and overcrowding, et cetera, presenting. So you'll see here the list of targeted populations in addition to the age-based groups that have been identified by government as the parallel group that will be going first in phase 2, hopefully in April or early May. Simultaneously, the idea of our province being very distributed as a population means that we have to look at very many clinics as well. So mass immunization clinics just mean a clinic that is meant for a whole community to drop in at, or a portion of a community. But they're also looking at mobile clinics, drive-through clinics in the early phases as well, and eventually getting into community clinics. So like we do now with influenza in our pharmacies. There's also going to be provision made for appointment-based clinics as well as drive-through and walk-in. And then, of course, as I said, as we get more vaccine supply, focusing on more sites, including those in the community with pharmacies, physician offices, or with places that have occupational health and safety nurses delivering for their employees. This is just an example of some of those places that have been identified so far. There was a bit of a trial run done with this year's flu campaign in the fall, trying to get more places getting used to delivering vaccines than we've seen in previous years. Of course, all of this takes people. And so, as I mentioned, we have a relatively small public health workforce. We can't deliver all of that on our own. Certainly not while trying to keep up routine immunizations as well. So a tremendous effort going into bringing back people who are retired, but also engaging other healthcare workers that can be trained to give immunization safely as well. So the other arm of this involves the public. We can be all prepared as a system, but we're really dependent on public acceptance and public demand. And the intentionality to be immunized against COVID-19 has really changed over time. We saw early on, a little less than 50% of people saying they had an intention to get immunized. By fall, that had actually fallen as people were starting to be influenced by distrust, I think, of messaging and government, fallen to around 40% in Canada. And then it rose again later in the fall to about 48%. But by the time we started seeing vaccine trials results coming out and governments approving them, that jumped up to now over 60% in early January. If you include those who say, I want to wait a few months and see how it works out for others, and then I'll get immunized, that jumps up again to 83% in Canada as of January. But I think one of the things we have to watch for is this potential for a side effect of the program, which is if we're focused on the most vulnerable and those that have the most serious impacts from COVID, we will see hospitalization rates and death rates come down, and maybe not as many people coming for testing. There will be a perception that we're out of the woods. And if that happens, we don't want to have vaccine demand go down. Because if we don't get this level of herd immunity, which different studies are pointing at anywhere between 60 and 80% likely that's going to be needed minimum, we will be seeing recurring waves of this over the next few years. So a lot of attention is going to have to be put on different types of social marketing campaigns targeted at groups that have low vaccine uptake and in trying to make sure we get enough people immunized. So I showed you this slide earlier in terms of some of the research that's going on, and I just wanted to bring it back because I think it's important to recognize that we have various types of research needed for pandemic control. And I know that there's probably an awful lot of PhD projects in the next few years that are going to be focused on dissecting this pandemic and how our response was. But in the immediate term, this importance of making sure that the university is presenting the evidence as it comes forward to decision makers so that they at least have the best evidence in front of them as they make these decisions, and that we are informed by the rates of immunity in the population over time as people have had both exposure to the disease and the vaccine to see have we reached that herd immunity level, and continuing to do vaccine acceptance studies and measures of adherence, et cetera, so that we can make sure we get out of this in the best way possible. So the final question I'll leave you with is just, I put in my title is the finish line in sight. And I guess you probably gathered by now, it depends what you consider the finish line. If what we're aiming for is 0 or very few deaths, we're going to need, of course, very high rates of coverage in phase 1, those in the early stages of phase 2, the most susceptible, and we need to get them immunized as soon as possible. And of course, the rest of us, while we're waiting for that vaccine to be delivered, we need to be at a much, much higher level of adherence with the public health measures so we can get the number of cases down and exposures to these people down. So a lot of provinces have responded to that with much more restrictive measures, at least in the short term, to bend that curve down much more quickly than we have here. That's what's going to be required for getting the deaths down. If our goal is 0 or very few ICU admissions and hospitalizations, we also need coverage in the rest of phase 2 and most of phase 3 as soon as possible. And meanwhile, again, still adhering to those public health measures in the meantime, because of that Swiss cheese model of exposure to the virus. It's going to take many months before we get to that level. But if our goal is actually 0 or very few new cases, well, that requires very high herd immunity, and not just in certain age groups, all subpopulations, and monitoring for reintroduction, and may require, as we heard, booster doses of vaccines in the coming years. And then lastly, I just say, is it good enough for us to have a finish line in Canada when we're dealing with a global problem like this? Really, we aren't going to reach that finish line until we've got this controlled at a global level, and that means assisting international colleagues as well. So I think just because we reach some of these targets locally or across Canada, we know the finish line isn't really in sight until we resolve this problem globally. So Canada needs to be a part of that global solution, and the University of Saskatchewan is doing its part to be what the world needs. Thank you.
Very much, Dr. Neudorf, for a very insightful discussion. I certainly learned a lot. I'm certain other audience members did as well. So we'll move on without delay into our question period, where we'll take audience questions. And so, Dr. Neudorf, I will bring the first audience question to you. What are your thoughts on double masking with regard to the new variants and future new variants?
Well, like many things, the studies are still being done. I think so much depends on what are the number 1 and number 2 masks that you're wearing. It's been shown already that high quality masks that are doing a good job at filtering are very effective on their own. As that quality or fit varies, then adding layers to a mask can certainly help. And so evidence is starting to emerge that for at least certain types of masks, a double masking may show more benefit in filtering virus particles than a single mask. But it really does depend on the type and fit of the mask that you're wearing in the first place.
Thank you very much. We'll go on to the second question. Should pregnant women and breastfeeding mothers get the vaccination?
Yeah, and this is something where, it's a bit of an issue of how we have to do drug testing and vaccine testing, that certain populations are excluded in those first tests. You don't want to expose certain individuals, those that are immune compromised or people who are pregnant, especially in early stages in those first studies. But invariably, what many of these studies show is that some people may not know they were pregnant when they got immunized. And so there are secondary studies done to follow up people that have been immunized and found out they were pregnant, and look at the safety there next. And then as you find out more, then they give more specific recommendations. So right now they're recommending caution in this area. But so far, there's no evidence to show that it's a danger to be immunized, whether you're pregnant or breastfeeding. But that's something that you'll be discussing with your individual healthcare provider, and it'll be changing as new evidence comes out with each of the new vaccines.
Thank you for that. I'm happy to hear that that'll be guided by evidence, the appropriate way to do it, not political decisions. I'll move on to the third question. Do you think Saskatchewan will have access to different vaccines as others are hopefully being approved, so that an individual will be able to choose the type of vaccine that they get?
So those are two questions, really, because yes, I do think that we already have 2 vaccines in the province. And as we find out if more are approved in Canada, 3 are undergoing that process to seek approval right now. We will likely see all of those make their way in different numbers. Some of it may be choice, but a lot of it will likely come down to what are the different vaccines good at. They may have different age profiles that they're licensed in, different effectiveness in different age groups or subpopulations. So you may see a discussion happening with a healthcare provider to best tailor which vaccine a person gets depending on their individual circumstance, versus others where if there isn't a particular need for them to get a certain vaccine, and encouragement to take whatever's there, obviously, that's offered to you at the time. But all of that is the type of information. It becomes more complex for people to make an informed decision. Each of these will have their own safety profiles and effectiveness, and making sure that first of all, we're licensing vaccines for the whole general public, but then if there's a specific need for certain people to gravitate more to one type of vaccine, that'll be something you discuss with the healthcare provider as well. Thank
you very much for that as well. We've got now a question. Can you please elaborate on how scientific and academic research, specifically forecasting, modeling types of research, how does that interact with the provincial government policy and the restriction planning?
Good question. So, like anything else, epidemiology has always informed what is potential or different types of scenarios that could take place depending on how the virus presents itself in your area. You learn from other places, you see how it unfolds there and how would that apply to our population. And we've done that with past pandemics as well. Computer modeling has gotten more and more detailed and sophisticated and has enabled us to take a lot more factors into account so that you can see different likelihoods now of different things playing out. And I think early on, certainly the modeling based on what people could see real time happening in other countries like Italy, early on Spain, made politicians take that modeling very, very seriously and put very, very strict restrictions in place. Since then, the fear of what's happening to the economy or jobs has meant that I think a false dichotomy has been put in place. We're looking at it's about economy or lives. Whereas on the other side, we've seen different types of government responses put in place to reduce that dichotomy and say that for short periods of time, what's the responsibility of government to assist businesses who are forced to close or people who've lost their jobs to have income in the short term. So I think, evidence is always brought forward to political decision making, and then the politicians need to decide which of that evidence they're going to listen to and where the balanced response is going to be. But it also then involves decisions on what types of political levers you put in place in addition to the restrictions to mitigate the impact on society. So I don't think it's really a false dichotomy, but always there ends up being this layered approach where evidence is brought forward and then politicians decide what to do with that evidence.
Thank you very much for that, Dr. Neudorf. So we are at the end or even past the end of the time for the question period, but I'm going to invoke the moderator's prerogative and shorten my own closing remarks because we have some more really interesting questions and I think the audience would appreciate those more. And so within the available I need a couple of minutes, so 5 more minutes, Dr. Neudorf. A few more questions for you. So we have an audience member who has written in, I have seen evidence that during the Spanish flu, Canadians were encouraged to wear masks and how to wear masks has been provided, through the media and how to make masks has been provided. So why was the medical community not encouraging people to make their own masks given the shortage of PPE? And yes, the education and information on risks of not getting the vaccine. Why is there not more of that? Sorry, that's 2 questions, but.
Mm-hmm. Yeah, I think, like anything, it's fascinating to look back early on and see just how little was actually known about how this virus spread and what was effective in stopping it. And a lot of previous research, especially in the English-speaking world, showed limited effectiveness of wearing homemade masks for stopping virus spread. Certainly medical grade masks were more effective, but there were shortages of those masks even for healthcare workers early on. So encouraging everyone to wear medical grade masks, of course, would have put the system in crisis as well. So unfortunately, we had to wait for the evidence to catch up to find out just how effective were some of these homemade masks. And as time went by, I think you saw messages from government changing about what they were asking people to do. And it's a bit confusing, but to me, I looked at it as heartening because what it meant was government was being transparent and saying, okay, well, this is what the evidence is now. Here's the limitations of what we know. And as we know more, we'll pass on more. So we've started to see a lot more detail that if you are going to make your own mask, what materials to use if you want it to be most effective, how to make sure the fit is good, the 3-layer mask with a polypropylene middle layer is best. Encountering misinformation about what the risks are, because people were thinking there were more risks to mask wearing than there actually were. And the fact, like anything else, health behavior takes time, and yet people adjust. The best example of that for me is kids. Everyone was so worried about how kids weren't going to be able to make this adjustment in school, and it didn't take more than a few days for many kids to just take it in stride that this is what they had to do. So, I think what we need to see from government is this transparency, this ability to change course as new evidence comes, and then try to get that education out to people as soon as possible. And then the public's need then is to make sure that we're going to reputable sites to see where are we pulling that information from, and who should we actually be listening to and believing here. And that's going to continue to be a challenge for us, as your second question is around good, transparent information about the various vaccine risks and side effects, and how we need to make sure people are aware about the safety profile and start coming to get the vaccine.
Thank you very much. So I'm going to try to slip in this last question, just a one-sentence answer if it's possible. Is September still an attainable goal for everyone to be vaccinated everyone who wants to be vaccinated in Canada to be vaccinated?
I think it is. You've seen the Herculean effort it's taken to get so many different types of providers there. As long as we get the volume of vaccine that's being promised to us right now, the system will be ready to respond, and I think it's still an attainable goal.
Thank you very much. With that, I'm going to call an end to the scheduled event and to ask all speakers to please turn on your video and your microphone. On behalf of all of our audience, I'd like to thank you, Dr. Gertz, Dr. Racine, Dr. Neudorf, in the order that you spoke, for bringing your time and your expertise to today's forum. I know that the audience has greatly appreciated it. I feel much more informed now than what I was before. And I want to extend that thanks on to all three of you for the continued efforts that you're going to do. We are still in this race against COVID, and we are not to the end of the line yet. And so I know there's going to be a lot more hard work going forward. And so you have the thanks, not only of the people in this session, but also of everyone in Saskatchewan. So thank you. Finally, I would like to extend my thanks to all the attendees of this year's Senate Forum, the race against COVID, and to inform you that there will be a recording of this session available on the university's website in case you have colleagues who were not available or were not able to attend today, but who you think would enjoy listening to the session going forward. With that, I am going to bid everyone a good, if frosty, Saskatchewan afternoon. Thank you, everyone.
Thank you.